Okay, I think we could start. So welcome everyone to this monthly seminar series organized by the ESAB-funded Special Topic Network called Integration of Speciation Research. So more information can be found on this network on its website, and from there, just a recap that people can also sign up to the network mailing list to automatically receive the program of the seminar series and other news. So today we will be starting a new series of 2 seminar sessions that will focus on the topic of coupling in speciation research. And with today a first session specifically on the contributions of multiple barriers to gene flow to the coupling processes with Erik, Dopman and Sophie Karrenberg as our speakers. So before I introduce our first speaker, maybe just a reminder on how the session will be run. So each speaker will talk for about 20 minutes, leaving a few questions after each talk for specific questions to each speaker. And then we will start a more general discussion session, staying on the same webinar link. So if you have any questions or comments after the talks or during the discussion session, please write them down in the chat, and Sean and I, who will be moderating this session, will ask your questions to the speakers. So now it's really a great pleasure to welcome Erik Dopman as our first speaker. So Erik is currently an associate professor in the Department of Biology at Tufts University in the States. So he started to work on speciation and adaptation already as an undergraduate student at the University of Texas in Austin, working on host plant adaptation in grasshoppers with Greg Sword, Larry Gilbert, and David Hillis. And he then did a PhD with Rick Harrison at Cornell University, where he initiated his work on the European corn borer moth by studying the genetics of reproductive isolation in this system. And before starting his own group, he moved as a postdoc at Harvard to work with Dan Hartle on genomic structural variation in fruit flies and yeast. So he's now leading a research group that tackles various aspects of speciation and adaptation, applying a diverse range of approaches from ecology to behavioral ecology, genomics, molecular genetics, to try to characterize at both the phenotypic and genetic levels barriers to gene flow, mostly using the European corn borer moth as a model. So he and his group recently published an article in Evolution on the consequences of coupled barriers to gene flow to the buildup of genomic differentiation. And Erik is also currently coordinating the writing of a section of the Cold Spring Harbor book on speciation that is dedicated to coupling. So of course, we are very happy that Erik, you accepted our invitation to give a talk today on this topic. So looking forward to listening to you, and whenever you are ready.
My pleasure. Yeah, thank you for having me. I will share my slides. Move my windows around. Yeah, so thank you very much for having me. So I'm really excited to talk about some of our research. I can move these windows around a little bit. There we go. Right. So I thought I'd start here by sort of having us think about the transition from one population into two species. And we, many of us at least, tend to imagine that the transition from one population to two species involves strong reproductive isolation. And if that's true, well, then understanding how speciation is completed really requires solving two interrelated long-term problems. The first problem is to identify the factors strengthening individual barriers to gene exchange. So, for example, the strengthening of something like pollinator isolation for a plant. But as many of you know, single barriers operating alone rarely contribute to strong reproductive isolation, right? So because of that, that generates the second problem. And that is to identify the factors causing different barriers to act together, to operate together, to couple. So, for example, if we were to add other barrier effects to pollinator isolation, we could imagine a population would transition to stronger reproductive isolation, bringing us closer to the origin of two new species. So, these factors that work together to bring barriers together are collectively referred to as coupling processes. It's a term that that has a relatively brief history, but I think it's increasing in prominence. So we can identify a general definition for coupling process as any process causing different barrier traits, barrier loci, or barrier effects to work together to produce a stronger overall barrier to gene flow. So this is a general definition that we came up with in a review article that we wrote. And as Carol mentioned, it's one of 6 chapters devoted to coupling in a new book on speciation that's to be published this year. So what are some examples of these coupling processes? So there are really no surprises here about the kinds of processes that might bring different barriers together, right? So we can imagine some aspects of selection could be involved, such as the reinforcement process. We can imagine many different population processes could also bring different barriers together, such as change in the distribution range, bringing barriers that are far apart in the distribution together in the same population pairs. And then of course, we can also imagine, lots of ways in which the genetic architecture can promote and constrain this process, right? So a good example would be pleiotropy, where you have single alleles that contribute to multiple barrier effects, in which case you have sort of simultaneous coupling of different barrier effects. So, there are many theoretical and empirical questions that remain to be unanswered about this topic, and I think that's one reason why I think it's a really exciting area to be conducting research in. But I will say that the long-term goal here is to both identify and establish the relative roles of these coupling processes. So, for empirists like myself in these empirical systems, the first necessary step to address that long-term goal is to document different stages of divergence that populations are in, and when barriers are in an uncoupled state, and when barriers are coupled together, right? So these would be patterns of coupling rather than coupling processes. So today, what I'd like to do is to share with you evidence of uncoupled and coupled barriers in the European cornborer moth, the Straenia nivalis, or the ECB moth. And a lot of the results that I'll share with you is part of a collaboration with Henry Kunerth and Jenny Kozak. Okay, so a brief background into the European cornborer, and I think this organism is kind of odd, or its name is kind of odd, because there are 4 words here, but 2 of them are true and 2 of them are complete lies, right? So let's talk about the 2 truths and 2 lies. Okay, the first word, European, that's a lie, right? So in fact, the species ranges from North America, where I am, to Asia, to North Africa, and Europe, right? Another lie, corn. So in fact, this species feeds on over 200 species of host plant, right? So it happens to feed on corn, and we happen to care about corn, so that's how it got that name. Now, the 2 truths. Indeed, it does bore holes through its host plants. You can sort of see that through these holes in the corn leaf. So it's a grass, right? So when corn grows, this is rolled up. Sort of like a sheet of paper, you can roll it up, and if you were to push a pencil through that and then open it up, right, you'd see these characteristic holes. And then the final truth, yes, it is indeed a moth, right? So two truths, two lies. So what are these uncoupled and coupled barriers in this species? So let's talk about that. So despite this really wide distribution, most of our work is done in New York State and in northeastern United States. In 2010, in that region, we estimated 12 potential barriers to gene flow between populations, and we could also quantify the strength of reproductive isolation among those barriers. So it's just a figure that summarizes those results. The most important ones are in green. The two strongest barriers to gene exchange is a temporal barrier and a behavioral barrier. So the strength of reproductive isolation is relatively strong. When you combine these two together, it gets even stronger. Reproductive isolation is about 0.9, so pretty strong. So I'm going to focus on these two in green, the temporal barrier and the behavioral barrier effects. And what I'd like to do is describe the phenotypic basis of the barrier effect. So we have barrier traits and the barrier effect. So in this case, the behavioral barrier effect is a consequence of divergence in the pheromones that populations use. So in this species, there are two pheromone strains referred to as the E and the Z pheromone strain, and they differ by what you'd expect, right? The pheromones that they use. So in this species, there are two kinds of females, and they release two kinds of pheromones. Females from the E strain, they release a 99:1 ratio of this two components pheromone blend. The Z strain makes the opposite ratio, a 3:97 ratio. And I should say that these two pheromone components are isomers of one another, E isomer and Z isomer, and that's how the strains got their name, the E and the Z strain. So those are the phenotypes of the males, right? So they release this long distance volatile sex pheromone. So males of the same strain, they prefer those pheromones. So the sons of, say, this female will strongly prefer that female sex pheromone blend, right? So there's strong attraction of those males, but very limited cross attraction of males to the opposite pheromone blend. And that's the basis of the behavioral barrier effect, is limited cross attraction. Now, what about this temporal barrier effect that I described? What's its phenotypic basis? So that comes from divergence in life cycle. So if we were to zoom in on at individual sites in New York, such as this one in Farmington, New York, and if we were to collect insects across the growing season, what we'd find in some sites is something like this. So in blue, what I'm showing you are all of the E strain moths that were trapped across the growing season. And what you can see is that there's an early season mating flight, and then there's a second late season mating flight. So 2 generations per year. We would call that bivoltine. So two generations per year bivoltine. And in orange, those are male moths that are from the Z strain. And those males they have one flight per year, and it's right in the middle of the year. So those are univoltine populations that are releasing and being attracted to the Z pheromone. So this life cycle divergence generates temporal isolation because adults don't live very long in the field. They live on average, 5 to 10 days. And so we're looking at, this axis down here showing us days of the growing season. Well, that means that males that are flying around here are going to be long dead when these females are trying to attract mates. And so that's the basis of temporal isolation. So at a location such as this, what we're looking at is a combination of life cycle and pheromones that differ between these population pairs. So this would be a bivoltine E and a univoltine Z population pair. But we don't see populations like this everywhere. So not too far away from Farmington is location where we have two populations that have the same life cycle, but they differ in pheromone, right? So this would be a case in which we have these two barriers have become uncoupled, right? So here we have them coupled together, life cycle and pheromone both differ. And here we have a situation where the populations only differ in pheromone, not life cycle. So I'm going to summarize a lot of data that were collected across years to sort of show you what we currently know about the kinds of populations that exist in nature. So there are populations that differ only in life cycle and have a temporal barrier. So that would be bivoltine Z and univoltine Z. I didn't show you examples of those. We have populations like those of Dresden, New York, that differ in pheromone and have a behavioral barrier, bivoltine Z and bivoltine E. And then we have population pairs that differ jointly in both of those phenotypes and have both a behavioral and temporal barrier. And those would be examples of coupling. So those barriers are uncoupled. So these results show us that there's geographic variation in the pattern of phenotypic coupling, but we can ask, does this coupling pattern also vary at the genotypic level as a buildup of linkage disequilibrium at the loci that underlie the traits, right? So we're asking whether there's a correlation between alleles at the underlying barrier loci. So I don't have time to really discuss the genetics in any detail, but I'm going to give a brief aside into one barrier locus because it relates to this discussion of coupling. And this is a study that was conducted with David Heckel and Astrid Groot and their laboratories. So theory predicts that linkage disequilibrium will build among loci underlying assortative mating, right? So a trait and a preference will tend to segregate together within populations, right? So that should build some LD between them at the underlying loci. So if that's true, then here's the question. Can we use the autosomal pheromone gene PGFAR, which was known, in a sort of genome scan of assortative mating to identify the unknown preference gene, which we knew was somewhere on the sex chromosome, but we didn't know where? So we set out to figure this out and to use this approach. And so how would this work? So if these assortative preferences cause assortative mating, well, then moths will tend to inherit E alleles at the PGFAR gene. That's the gene that determines the pheromone blend, as well as E alleles at this unknown preference gene. Or individuals will tend to inherit Z alleles at PGFAR and Z alleles at this preference gene, right? So we can evaluate this by quantifying and measuring LD between PGFAR and all SNPs on the sex chromosome, on that Z chromosome. So we are expecting that LD may build up between these two loci. And that should be a signal that we can use to identify the preference gene. So just to make this clear, here I'm showing us the association between alleles at this PGFAR gene. We have two E alleles, an E and a Z allele for heterozygote, and two Z alleles. And then sort of imagine this is the preference gene, right? So if this is the preference gene and assortative preferences cause assortative mating, maybe we would expect to see some kind of association between the alleles at the two loci. So higher squared values there. Whereas for some random gene, that's, perhaps there be no relationship between the two. Does that make sense? So we're looking for this pattern. So we did that. We quantified LD using R squared between amino acid changes at this pheromone gene PGFAR and all SNPs across the Z chromosome, this 20 some odd million base pair chromosomes. So what did we find? So here I'm showing you all of the SNPs across the Z chromosome, lots and lots of variation. But notably in the 18 and 19 megabase region, there is a jump in associations. So just to be clear, this is the sex chromosome, and this is an autosome, so chromosome 12, right? So, but interestingly, the association is comparable to what you'd expect to see with tight physical linkage. So if we were to zoom in on this region, what we find is that the SNPs that have the strongest association are within the gene bric-a-brac. And I should note that bric-a-brac was identified using other methods, so we felt pretty confident that this was it. So if we were to zoom in on that particular SNP right there, that's the SNP that has the highest association. Here's what we found. So these are empirical results. So here's PGFR genotype with different alleles, bric-a-brac genotype with different alleles. And here we have a strong association, the R-squared value of 0.71. This is sort of what you'd expect for a typical gene on the Z chromosome, so relatively low R-squared values. Okay, so just to summarize all that, of the decade worth of work, so we've identified two loci that together underlie life cycle divergence. They're both genes in the circadian clock pathway, the circadian clock gene period and pigment dispersing factor receptor. We described those in Kozak et al. 2019. And then we have these two genes that underlie pheromone divergence, bric-a-brac and PGFR, the fatty acyl reductase, described in these two papers. Okay, so with this data, with this information in mind, we can then come to this question that I asked you earlier, right? So this coupling vary at the genotypic level as a buildup of LD. So we evaluated this question by using R-square again, except this time we're using PGFR, right? The same gene I described earlier. That's on autosome 12. It underlies the female pheromone differences. And PDFR, this is one of two loci that underlie differences in life cycle. It's on the sex chromosome. So again, a loci on different chromosomes. And we quantified R-squared values between populations that differed in pheromone alone or that differed in pheromone and in life cycle. And what we'd expect is that there'd be higher R-squared values in populations such as this, right? So what do we find? Exactly what you might expect, right? So there was a sort of four-fold increase in R-squared in populations that differed in life cycle and in pheromone, right? So we're seeing evidence of the coming together of these barrier effects at the phenotypic level, as well as at the genotypic level, right? So there's the question, and there's the answer, yes. Okay, so the final question I'd like to tell you about is this one. Does the buildup of LD among multiple barrier loci, right, which I just showed you, does that enhance differentiation at those loci? And does that buildup of LD among multiple barrier loci also enhance the barrier to gene flow in the rest of the genome, right? So differentiating between the barrier loci themselves and everything else. So it should, right? So based on the organismal estimates of reproductive isolation I described earlier, which ranges from 0.65 all the way up to 0.9, it should. Right. So having said that, as many of us are aware, right, expected patterns of differentiation at barrier loci are very much idealized, and they may not reflect the realities of nature for all kinds of reasons, right? So caution is warranted. So having said that, what do we find, right? So, okay. So I'm going to show you a number of figures that show us the levels of differentiation as measured by FST as a function of genomic position, and we'll be looking at barrier loci separately from the genomic background. And then I'm going to be showing you differentiation in populations that are 4 different categories. So population pairs that don't phenotypically differ in either one of these phenotypes. Populations that differ in life cycle alone and have a temporal barrier in green. Populations that differ in pheromone alone and have a behavioral barrier in teal. And then in purple, those would be population pairs that differ in both phenotypes and have coupled barriers. Okay, so let's look at what we found for the barrier loci themselves, right? So we're looking at the genes that underlie the traits which cause the barrier effect, right? So over here on the right, we're looking at the preference gene bric-a-brac. So populations that don't phenotypically differ and that only differ in life cycle have very limited genetic differentiation at the preference gene, exactly what you'd expect, right? These populations don't differ in the phenotype. You wouldn't expect differentiation at the genetic level. But we see a rise in differentiation when populations differ in pheromone. Again, it's exactly what you'd expect, right? You're looking at the locus that underlies the trait. So we expect to see strong differentiation. Well, what's really cool is that when you add another barrier in the background, you see differentiation increase even further, right? So there's this increase, this elevation of differentiation at bric-a-brac. And you see the reverse pattern at the life cycle loci. So there's a combination of period and PDFR. So differentiation is initially relatively, it's moderately elevated for populations at different life cycle. But when you add differences in pheromone, FST increases, right? So it's possible that this is evidence of some kind of indirect barrier effect from other barrier loci. So the idea here is that LD among barrier loci allows for indirect selection. And so therefore, overall selection at each barrier locus is strengthened. And so we're seeing, these genes sort of being pulled apart. So we answered this question. So differentiation does appear to be elevated at the barrier loci. So what about the barrier gene flow and the rest of the genome? What does that look like? So here we're looking at the genomic background. We're removing all of the scaffolds that harbor the barrier loci. And we're looking at the same four categories, right? So populations that don't differ at all, different life cycle alone, different pheromone alone, or differ in both life cycle and pheromone. So there's a lot of information, but I sort of summarize the main take home here on top. So most of these chromosomes, they show little genetic differentiation. It doesn't matter how many phenotypic differences exist between the populations being compared. But, it's pretty striking that there are two outliers. The two chromosomes that appear to be outliers are the two chromosomes that harbor the barrier loci, right? So that's sort of what you'd expect. Chromosome 12, well, that's the one that houses the pheromone production gene PGFR, and the sex chromosome, the Z chromosome, it has bric-a-brac, the preference gene, and then both life cycle genes, period and PDFR. So if we were to zoom in onto those two chromosomes, the Z and chromosome 12, what do we find? Well, we find that chromosomal differentiation, it increases as the number of barriers accumulates, right? So exactly sort of what you might expect, and it's similar to the kind of patterns that we looked at with the barrier loci. But you'll notice that the Z chromosome is a very different pattern compared to chromosome 12, and it might be that is because that chromosome has 3 barrier loci on it, whereas the autosome chromosome 12 only has 1. So that might be a so-called large Z effect, or sometimes referred to as a large X effect for other organisms. And I will note that chromosome 12, right? So this chromosome lacks any genes underlying life cycle. And yet, when you have a chromosome 12, FST increases under joint divergence in purple. And that might be additional evidence of this sort of indirect barrier effect, the consequences of LD with other barrier loci are having an effect on this completely physically unlinked chromosome. Okay, one final result that I'll share is just another perspective of what is essentially the same kind of patterns. We're looking at only the Z chromosome now and only collinear regions of the Z chromosome. And in 3 kinds of populations, one that doesn't differ in either pheromone or life cycle, populations that differ in pheromone, and populations that differ in both pheromone and in life cycle. Right. So, I mean, the pattern already is very, very obvious. But if you look at the number of individuals that show evidence of admixture that said intermediate levels of co-ancestry, you can see that about half the population has mixed ancestry when a single barrier exists, right? So in green, whereas only 3 show evidence of mixture when barriers are coupled. Right, so it does seem to be. It seems like these coupling of the barriers is allowing, 2 gene pools to occur, at least for these markers. And interestingly, the levels of admixture when barriers are coupled are not statistically significantly different from the levels of admixture when populations lack any barriers at all. right? So there are no differences between these two populations in terms of the numbers of admixed individuals. So the answer to the second question, does the buildup of LD among multiple barrier loci enhance the barrier to gene flow in the rest of the genome? Not genome-wide, but definitely on certain chromosomes. And again, caution is warranted, and I think there are some aspects of population processes that we're going to want to take a closer look at. Okay, so with that, just let me give you some take-home messages. I argue that understanding the role of coupling during speciation requires documenting coincidence of barrier effects in populations at different stages of divergence. At least in the Kornborg system, it shows evidence of patterns of coupling across these organizational levels, right? So I showed you evidence of phenotypic variation in patterns of coupling with a buildup of barrier effects. I showed you evidence at the genetic level with a buildup of LD among the underlying barrier loci. And I showed you evidence at the genomic level with a buildup of chromosome-wide LD. So with all of this in mind, the next step for us is to establish the relative roles of these factors that are generating those patterns, right? So what is it about, could selection be involved? Could population processes involved? What's the role of architecture in promoting and constraining, these barriers from coming together? So that's our next step. And finally, I thought I would end here with my perspective on the benefits of coupling as a concept. So from my perspective, there are two main benefits. Number one, it sort of fills a need to more fully describe this transition from differentiated population to new species through the buildup of reproductive isolation. So much, there's been a lot of focus and attention on the strengthening of individual barriers. And I think what this does, adoption of this concept, there's a second benefit. It increases emphasis on the second speciation problem, right? So how do combining multiple barriers allow for strong reproductive isolation and therefore allow speciation to complete? So with that, I'd like to thank my collaborators, funding sources, and I'm happy to take any questions. Thank you.
Thank you very much, Erik, for this great talk, inspiring. Would anyone has a question or two to Erik? We may have time for one or two questions. Maybe I can start with just one, but it's a kind of general one. But so you explained that you have identified many more barriers to gene flow in your system that may contribute only moderately to reproductive isolation, but still they exist. First, do you think you are still missing some in this system that you haven't identified yet? Like the role of hydrocarbon, cuticular hydrocarbon in sexual isolation, for example, or something like that. And second, do you want to tackle coupling among all of the barrier effects, and is it feasible?
Yeah, so the second question first, it depends on how long I live and how much money is available. But I think the first question, I think that ideally, that would be fantastic, right? So, I mean, I'm sure that there are relevant phenotypes and barriers that that we've completely missed. So, what I've chosen to do is, I said early on that I would focus on the ones that empirically were the strongest, thinking like I'd have the greatest chance of detecting an influence of those. So, having said that, I think it's possible. So one hypothesis is that some of these barriers are a consequence of pleiotropy. So in particular, the temporal barrier that we're observing across the growing season may be influencing the temporal barrier that we observe across a 24-hour period. And now, if you remember that the two genes we identified are both involved in the circadian clock pathway, which are known to control circadian rhythm, including timing of reproduction across a 24-hour period. So we have ongoing work to evaluate the possibility of pleiotropy. If that's true, then we may already be studying the basis and the consequences across more than the two barriers that I described.
And related to that, sorry, I'm following up, but because you would like to address the processes underlying this coupling pattern that you see. Do you have the feeling that you have ingredients for reinforcement, for example, like, because I'm not aware of post-zygotic isolation, but maybe.
Yeah. So. Possibly. So one of the questions that we will want to address is the possibility that there is some multiple effect traits that we're investigating. So these would be traits that have an influence on more than one component of isolation. Mm-hmm. So for these two traits, for example, the consequences of having an intermediate pheromone preference, or an intermediate timing of seasonality might be pretty bad. It might be right. So I think that's one possibility where you could have the reinforcement of these two barriers together, because if each of them contributes to some kind of underdominance in hybrids, then it's possible they could be mutually reinforcing one another.
Cool.
But we have to test that. We have to evaluate. Yeah, thanks.
Okay, so maybe we can switch to the second speaker, and we'll have more time for more questions later.
Okay, great. So in that case, it's now my pleasure to introduce our next speaker, Sophie Karrenberg. So Sophie obtained her PhD from ETH Zurich in Switzerland, and then undertook postdoctoral research at Indiana University in the USA, followed by a second postdoc back at ETH Zurich. And so Sophie is now a group leader in the Department of Ecology and Genetics at Uppsala University. Her group's research program focuses broadly on understanding the ecological and genetic mechanisms that contribute to lineage divergence. And they mainly study a variety of plant systems, including Silene, Arabidopsis, and Salix. And her group's recent work on speciation in the plant genus Silene has focused mainly on dissecting the architecture of different reproductive barriers. And so this includes understanding how different reproductive barriers contribute to the speciation process individually, how they're controlled and coupled genetically, and whether their effects on gene flow can be detected using genomic data. And one of the things that I think really stands out about research from Sophie's group is the effort that's made to really combine ecological approaches, such as really large transplants and crossing experiments, with genetic investigations, including methods like population genomic analyses and demographic modeling. And so with that, I'd just like to hand over to Sophie. So feel free to share your screen.
Thank you very much for the introduction and for having me here. Just a second. Sorry, I have a small issue with my screen soon coming up. Sorry, now it's better. Sorry. So, thanks for inviting me here. I'm happy to talk about my research, and I've been trying to go through my results, trying to find out what I have to say about coupling, which I didn't explicitly study like Erik before me. So now I am in Uppsala, and sometimes we do get some northern lights, just in case you don't know where we are. Right, so when Erik and I met before the seminar, we thought that, well, I'm studying more the late stage speciation, whereas he's studying an earlier phase in the speciation process. And we can think of it like this, that as divergence increases, gene flow is reduced, right? This sort of process. And when we're down here, we would talk of late stage speciation. So we have a strong separation, but still ongoing gene flow. And usually we have a complex array of reproductive barriers, ecological divergence and assortative mating often. The difficulty is, of course, to know when they arose. And in a late stage speciation case, we're not so sure about that. And then it is also very common that these involve very complex traits with complex architectures. And now let me jump right into our study system. So we studied two species of campions, Silene dioica and Silene latifolia, and these are perennial plants that have separate sexes. So they are dioecious, and they have a very large genome, which has led to troubles. But finally, now we have a draft reference together with others. I cannot report on that today because we just got access to it, actually. These two species differ in habitat. The red flowering one occurs in more moisture environments, and this one in more drier environments. And then they also occur at different elevations, and they have different pollinators. All of these differences are not completely excluding the other species. So, and this is an important aspect, and this is one of the reasons why we still do see gene flow, I would say. Right, so my previous doctoral student, Xiaodong Liu, and we did a couple of genetic studies on these two species, and here we have their distributions, the white flowering one going much farther into the east, but we also see a very large area of overlap. And of course, one basic thing you can do is to do a structure analysis where we see, well, the two species are really separated, but there is some admixture going on, right? Here, the red dioica indicating color going into the latifolia populations and vice versa. Another important thing to think about is the demographic history, because it kind of really matters whether you have barriers arising as kind of divergence peaks, or whether you have divergence peaks due to the erosion of divergence in other places, which would happen in secondary contact. And that's why we undertook an analysis of the demographic history, and we could actually confirm that the most likely scenario was divergence with gene flow. And this puts kind of a different perspective on understanding barriers and their effect on divergence. We also were able to figure out that there is some part of the genome that seems to evolve without gene flow. And these areas or regions are very interesting because these could be the very barrier regions we're interested in. The overall FST is 0.28 here, and this is indeed much higher than the example we've just seen from Erik, where it was more 0.05, if I got that right, for the non-barrier chromosomes, right? All right. And we have looked at mechanisms of reproductive isolation in this system for a long time. And let me just briefly summarize what we see there. So we see a large number of different barriers, all of which are incomplete. And here I have calculated in this Chen and Sobel methods the so-called cumulative reproductive isolation, where barriers add up onto each other in the order they occur. And this is reproductive isolation from the white flowering latifolia gene flow into dioica. And here we see a moderate effect of geography and a large effect of ecological differentiation. There is also some flowering divergence and assortative pollination, but clearly these barriers can only take effect when actually the two species co-occur. Otherwise, it doesn't really matter, right? There is also conspecific pollen precedence. So that is when the own species pollen kind of wins a race, and a number of post-zygotic barriers, all of which are ecological, as I will show in a moment. The overall barrier strength is 0.98, so this is very high. And we see that the ecologically influenced barriers are by far the strongest. So one of the experiments that we did to figure out about the adaptation of these two species was a big crossing array, where we cross the two species with themselves, so to say, different from different populations, and two generations of crosses also between species. So we end up with second generation crosses within species F1 and F2. So these are recombinant progeny, and then the other species. And then we went on to transplant those crosses into 6 different sites. within 3 populations of each species. And this was done by Adrien Favre. And this is the main result of this study here. So here we see something we called cumulative fitness. It actually is the number of years the plant flowered. It integrates survival and flowering, so reproduction. And oh, I think I'm missing the color code. So let me, no, it says here, let me walk you through it. So in red, it is Silene daiica, and we have the Silene latifolia sites on top here. And white is Silene latifolia, SL. So if we now compare red and white here, we see that the foreign species, daiica in this site, always underperforms as compared to the native species. And this is true in all 3 sites. When we now look at the daiica sites, we see the reverse. The native species, Silene daiica, outperforms the foreign species in all 3 cases. We can further look at the hybrids. So in blue here, the F1 hybrids. And I made this line as to what is the expected value, the average of the parents here. And the F1 doesn't really differ from the expectation, maybe a little bit here. But the F2 hybrids, they differ quite massively from the expected value here. So this is something one refers to as hybrid breakdown. So in the F2 hybrid, they really don't do too well in these habitats. So we have strong evidence for adaptation in this system. And now I would like to go on a little bit to talk about the difference between the sites. So if habitat adaptation now drives this divergence and reproductive isolation, we could expect in sites from weak adaptation to strong adaptation, that in these sites also the selection against hybrids increases and the hybrid breakdown increases. So this would be evidence for ecological divergence kind of driving this system. And we can look at it, we can plot the strengths of habitat adaptation against the selection against hybrids. And we see that this really increases across sites in both types of hybrids. And this means that the hybrid underperformance is due to the adaptive differentiation and not to intrinsic incompatibilities, because in that case, it shouldn't increase in this way. So this is an ecologically dependent post-zygotic reproductive isolation here. And when we look at the hybrid breakdown, we see a similar pattern. So breakdown here is the percentage reduction as compared to the expected value. And it's really quite massive in the site where daiica had the strongest evidence for adaptation. So another way to plot it is actually to calculate the reproductive isolation index across sites. And this is what we see here. So here we have the 3 daiica sites, and then we have the reproductive isolation index for a selection against migrants, the other species, and the 2 hybrids. And I think what we see here is actually coupling. So we see coupling of these pre- and post-zygotic barriers across the sites. So at a site where we have a very strong barrier against incoming migrants, we also have stronger barriers against hybrids. So I would refer to this as coupling. I will learn from you whether you agree with that or not later on, I suppose. But another thing we see here also, as with all ecological barriers, well, they are kind of not very constant, right? They vary with the environment. And this is important when you measure any effect strengths here. It's all a bit wobbly, I would say. Right. So then we also went on to study the genetic architecture of our fitness proxy in this experiment. So what we did is we took the F2 hybrids from 2 sites per habitat, and we applied reduced representation sequencing and used these Bayesian sparse linear mixed models that can account for family structure and allow for polygenic architecture. And the overall result here is, so we did that separately for the habitats, is that the percent variation explained is 14 and 12%. and that the number of variants with effects on fitness here on our fitness proxy is rather high. So between 40 and 50. Okay, that's not unexpected for such a complex trait. All sorts of genes can contribute to that. But another thing we can look at is the actually effect size. So this is the allelic effect per allele, and this is referred to as beta hat. And we want to look at the effect in one habitat, here is a latifolia habitat, against the effect in the other habitat to try to understand where do these alleles come from and how does the system evolve. And let me walk you through a little bit what we can expect to see. So one pattern that we could expect is antagonistic, it's a T missing, sorry, pleiotropy. So here we would have positive effects in the latifolia habitat coupled or at the same time with negative effects in the other habitat, right? It's good in one habitat and bad in the other. So you get this diagonal. And then if we also think about where the alleles are coming from, so this is not the data, it's the expectation, right? We would sort of expect that the alleles that are beneficial in one habitat actually come from that species. So in blue here from latifolia. And I express that here as the allele frequency difference, where blue indicates the allele is more common in latifolia and red more common in daiica. So this is one of two patterns we could expect. The other one would be conditional neutrality. And here we would expect that we have positive effects in one habitat with no effect in the other habitat. So the same allele has no effect in the other habitat. And same here, it may increase fitness in the daiica habitat, but has no effect in the latifolia habitat. So basically a cross pattern of some sort. And let's look at our results. So these are the actual results from our study. And this conforms more to a conditional neutrality pattern. So we have more variation here around 0. And the other thing we see, and we were ourselves quite struck by this, is that we have latifolia-derived alleles that increase fitness in the latifolia habitat. But what we also see is daiica-derived alleles that decrease fitness. So this is kind of more difficult to explain because when you have a lot of gene flow, you would expect purging of deleterious alleles. But here we have a situation where they arose probably in the daiica habitat and where they have no effect. So it's hidden deleterious variation. But when these variants are moved to the latifolia habitat, they have a strong negative effect or various negative effects. And if we look in the daiica habitat direction, the effect is not as strong, but it's also more red here and more blue here, right? So the thing we found, conditionally positive effects tend to come from the local species and conditionally negative effects tend to come from the foreign species. And given that these deleterious effects can only kind of stay if there's no gene flow, I think that this might also have to do with coupling or maybe rather feedback, because this can only arise in the situation where you already have strong barriers. Otherwise, these variants wouldn't persist. If you had tons of gene flow everywhere, they would get purged. So I think that this kind of coupling among any barriers strengthens the reproductive isolation and divergence, and it leads to selection against foreign alleles, further increasing divergence. Before we did this, we also had done a study in controlled condition, and this was a classic QTL study where we used 2 different F2 families, and we tried to figure out a number of traits that were associated with assortative mating, ecological divergence, phenology, and also male fertility reduction. And what we found is really quite complex. So here you see the linkage groups, 12 linkage groups of Silene, and these are F2 families again. And then in the different color, you see QTLs that we found in these different categories, and the empty and full symbols are the two crosses we used. And the one thing we see here, it's kind of everywhere. So these traits that are associated with reproductive isolation can be found throughout the genome. And you also see kind of that, well, sometimes you find an empty and a full one of the same trait next to each other, but a lot of times you don't. And we analyzed that a little bit further, and we came to the conclusion, well, this probably is a sign of a redundant genetic control, where you have many more loci than you would find in any one cross, more loci segregating in the populations. And then the third thing that we may see here as well, it looks like they are kind of always close together, right? The QTLs. But you also see the very large lot intervals. So this was done in 18, so it's a while ago. But we did try to simulate, right? Are the differences, the distances, linkage map distances between QTLs smaller or larger than expected? And in fact, here we have a little bit the other way around. The proportion of distances below 2 centimorgan in simulated randomly placed QTLs is actually much smaller than the observed proportion of distances below 2 centimorgan. And this does indicate that based on these results, it looks like coupling, but there are, of course, a couple of other things that could be going on, like finding more variance in the middle and things like that. But it looks like it. And why could that be? It's hard to imagine there would be selection for coupling or things like that. It's just too complex and too redundant, I think. But another thing that we believe that might be going on is to do with the genomic landscape of divergence in this system. Another thing we did is that we looked at our linkage groups. Soon we will have the reference, yes, but we looked at the linkage groups and we looked at hierarchical FST between the species. And here I highlighted kind of very high divergence outlier loci that had very high FST. And what we see that it's always kind of in the middle of the linkage group that we find a peak in FST. And this is an expected pattern, and it depends on recombination rate variation or on just being in the middle and not getting as many crossing over events. So we could also find that these outlier regions have elevated sequence divergence and reduced pi, might be consistent with selection. And another thing we did is that we looked at isolation by distance patterns in the outlier loci up here, and we find no pattern. And these are the low divergence loci. And there we do have an increase here with distance. So this is across the set of populations that I had shown earlier on the map. This effect is maybe not super strong, but it is there. And we think that this could also be a way to empirically identify barrier loci that actually don't show signs of gene flow. So in a way, we think that coupling could occur just through preferential establishment of reproductive isolation loci in these low recombination regions. I think this could be a rather strong effect in cases where you have this super complex genetic architecture, where you have loci with minute effects contributing to isolation, and we have these loci all over the place. And they are just more likely to be retained in these low recombination regions. Yeah, and based on our results, we think that we do have such a situation where you have a very large number of loci contributing to reproductive isolation. Right, and in conclusion, in this late-stage speciation, we have a couple of things that pertain to coupling. So the first thing is that we seem to see evidence for coupling of pre- and post-zygotic barriers across sites. It's kind of a non-standard way of looking at it. I know, please criticize me later. Then we find adaptive divergence through what appears to be conditionally neutral alleles. And we feel it could be that there's a feedback going on, that especially these conditionally negative effect alleles can only arise when there's reduced gene flow. And thirdly, we find a complex architecture of reproductive isolation traits, and we propose that those might arise preferentially in low recombination regions where we have evidence for a rise in FST, and also from demographic modeling for loci without gene flow. We just don't know where they are. That's the nature of these demographic modeling things. And with that, I would like to thank my collaborators, Alex and Sylvain, and PhD students, Xiaodong, Adrien, and Joël, and also my postdoc, my previous postdoc, Susanne, and master student, Emily, and the funders for providing funds for these projects. Thank you very much for listening.
Okay, thanks very much, Sophie. That was a really great talk, and you've got some really stunning data sets there. We've got time for a couple of questions before we move into the discussion, if anyone has any. So Roger's got his hand up. Roger Butlin.
Hi, Sophie. I'm just wondering whether you see much evidence for transaggressive segregation in your F2s, and whether you think that might be contributing to the reduced fitness of the F2s. It's sort of thing you might expect where you have this complex architecture and alleles working in both directions.
Yeah, that's we do see some evidence for that, but I always think there is a difference whether you look at performance or fitness or something related to that very closely. Of course, early death could be seen as transgressive, but it could also just be, low performance. And then traits like flowers and so forth, yes, we do see that. But as expected, we also see transgression in the other way. For example, we always found individuals that we referred to as a salad that would get that big, a rosette, but they usually never flowered, so they were not too successful, actually. Yeah,
thanks.
Great. Any other questions? So I might ask one if there's no others. I think Liz, is it Liz Gortado has a question? Yep.
Yeah, thanks, Sophie. So I thought it was really interesting that you found selection against migrants in the same habitats where you have selection against those F2 hybrids. Do you think that's the same ecological mechanism generating that selection? So the reason that migrants are unsuccessful at establishing is also why the F2s are unsuccessful, or do you think those are different factors?
I think they might be the same. So I didn't go into detail here, but there also is the possibility that there is some sort of intrinsic effect that gets stronger when the habitat is challenging. And in the paper, we also did a graph with that, and there also was a pattern, but it wasn't as strong. So we cannot exclude that intrinsic effects that just get worse. The worse the habitat gets, so to say, those could also contribute. And we don't have direct evidence that it would be the same alleles, actually. So for this, I think genetic evidence would be needed. Yeah. It is quite likely, though, right? Otherwise, we wouldn't see this strong correlation. But yeah, no, we don't have that level of detail. How would you do it? It's difficult, right? So we I would say we took a little bit of a shortcut there, right? We focused on the fitness proxy directly and didn't go via the traits here. It was too tedious, too much work, too many traits, so to say. So we first wanted to focus on the fitness proxy itself, and therefore I can't really answer what trait might have mediated that. Probably some physiological or root trait where we had evidence, like, you want to dig out 1,000 roots per site. Yeah.
Okay, great. I think in that case, we're 5 minutes past 6. So I think we'll move on to the discussion session. Before doing that, I just want to thank both of the speakers for really fantastic and stimulating talks, which should definitely lead to a stimulating discussion. So thanks, and I'll stop the recording there.